Yeah, well, he's sitting up here. He's the Shelby Foote of the accounting community. He doesn't like that. But he is he's got the look, he's an author. I'll let folks know. And at the end, when they're done around the Q&A, they're going to wrap it up with a mortuary brief. Lieutenant Colonel Laura A. Reagan, United States Air Force, is Director of the DoD DNA Registry, Armed Forces Medical Examiner System. In this role, she oversees both the DoD DNA Repository and Armed Forces DNA Identification Laboratory, AFID. A forensic anthropologist by training, she's a graduate of the United States Air Force Academy and holds a PhD in physical anthropology from the University of Florida. She has performed multiple casework around the globe, including a multitude of military blast events, over 40 air mishaps, the Polk and air crash outside Buffalo, New York, the mass shooting in Fort Hood, Texas, and the response to the aftermath of the Haiti earthquake. Lieutenant Colonel Reagan serves as a founding member of the scientific working group of disaster victim identification. Next to her is Dr. Tom Holland, the scientific director of the Joint QWMI County Unit. He began working there in 1992 when it was with the U.S. Army Central Identification Laboratory in Hawaii, or SILHA. He has supervised recovery excavations in China, Korea, Vietnam, Cambodia, Laos, and Iran. Dr. Holland received his Bachelor and Master of Arts degrees and his doctorate from the University of Missouri, Columbia, Missouri. He's a certified diplomate of the American Board of Forensic Anthropology and a member of the American Society of Crime and Laboratory Forensics. He's widely known as a raconteur extraordinaire. Dr. Reagan, you're up.
Thank you. Well, good afternoon. Welcome back from lunch. Mr. Panic is here today. He's signing autographs out there, and they allow me to speak out here, so I do appreciate that. Before I get started, my organization actually makes me state that while the facts are on these slides, anything I say is my opinion and that of mine alone, so it does not necessarily reflect the United States government or the United States Army Medical Research and Material Command, which is my parent unit. So if I say anything, just take it as it's my opinion. What I'm going to do is I'm going to talk to you a little bit about our organization in general. A lot of folks don't know that AFDL does not belong to JPAC or DPMO. We're our own independent organization, and we fall under the Armed Forces Medical Examiner System. So I'm going to talk really briefly about that, talk about our division in general, and then I'm going to be going into the DNA science. I know a lot of you have been to these briefings before, so you've got a basic understanding of DNA technology, but it's always changing. So we're going to talk to you about the basics and talk to you about some advances. Now, originally, I thought I was going to go after Dr. Mullins, so I've got some case examples too that he's going to speak to in more detail, but we'll talk about how DNA contributes to the identification process. And then also talk about some things that are on the horizon as far as DNA technologies go. We belong to the Armed Forces Medical Examiner System. The Armed Forces Medical Examiner System is the only medical examiner system in the federal government that caters to the Department of Defense and all other federal agencies. So if any of you are fans of the X-Files, there is no Scully. We would do the IDs, we would do the autopsies if there were any FBI cases out there, perhaps aliens that had landed. So a lot of you know what a medical examiner is from television. Quincy is one of the earliest ones. A lot of things. Ducky from NCIS. We don't work for law enforcement, we're our own independent agency. But one of the differences between the Armed Forces Medical Examiner System and JPAC, and SIL specifically, is that we tend to deal, the Armed Forces Medical Examiner System, with current conflicts. So all the folks that are coming back from Afghanistan and from the recent conflict in Iraq, they all would come through our office in Dover Air Force Base. And that's where we would conduct autopsies on them. And we do this, we know they died in combat. And a lot of people ask us, well, why would you do an autopsy on a case that you know what killed them? Well, we do this for a multitude of reasons. Let me give you two words: Pat Tillman. If anybody knows the Pat Tillman case, it's because we conducted an autopsy on him and we found green tip NATO rounds when we were looking for evidence. So evidence of friendly fire. But we also want to know specifically how our combat troops died so we can prevent similar deaths in the future and mitigate injuries in the field. So we actually work with other medical professionals who treat the wounded. We work with the individuals who design body armor and the vehicles to protect our troops and hopefully save lives. Additionally, we're very proud of the fact that we have been able to identify all fallen service members from the Iraq conflict. The only person that we are unaccounted for as far as Afghanistan goes is Bowie Bergdahl, and we're currently working very hard to try to locate his whereabouts in Afghanistan. So our organization, the Armed Forces Medical Examiner System, as you can see, we are one of 4 divisions. So we've got the medical examiners, we have forensic toxicology, they do all the drug testing, you've got the DNA laboratory and the repository that I'll go into in a little bit, and then we also have epidemiologists. When you hear them talking about, for instance, suicide statistics in the military, they're the ones that are compiling all the data on how all of our active service members are dying. As far as the DoD DNA registry goes, this is our vision, and basically we just want to be the best. We are the only human identification laboratory in the Department of Defense. There are other DNA laboratories that perform law enforcement work, criminal work, but we're the only one dedicated to identifying human remains. And that's very unique. And we do this through 2 different branches of our division. Basically, we're comprised of both a service laboratory, that's AFDL, that's what most people are familiar with, but then we also have the Armed Forces Repository for specimen samples for the identification of remains. That's quite a mouthful, isn't it? It's the repository. It's where we store all of our active duty service members' blood reference cards. And this is what makes these identifications for current conflicts so different than the past conflicts, because we have the references on file. We don't have to go to the families in most cases in order to get these. Now, this organization, and AFDL in general, we have a variety of different missions. We support not only past accounting and human identification efforts, but current conflicts as well. We also support intelligence agencies working downrange to assist in tactical decisions that are being made on the battlefield. We assist in law enforcement cases to a minor extent, primarily for paternity testing in criminal casework, because our DNA technologists are very adept at kinship analyses. Obviously, if we're going out and we're getting family references from all of you, they're very good at looking at family relationships and seeing how people are related, so they can tell if 2 individuals are actually the parent of an offspring. And we can use that. We do testify in court cases because of that. And additionally, we respond to humanitarian missions. Hurricane Katrina, for instance, we sent teams out there. The Colgan air crash up in Buffalo, we assisted in the identification of those decedents as well. So the repository currently has almost 6.7 million DNA reference cards on file to assist in identification of our service members. There are civilians that are represented there, the Coast Guard is represented there, and some other federal agencies. This is Mr. Larry Drayton, he is our division chief, and what you can see, we just collect a few spots of blood. So anybody who served after about 1995 should have a DNA reference card on file in our repository. There's only one repository in the entire Department of Defense, and that's located up at Dover Air Force Base. AFDL, the laboratory, has a variety of different functions, and I already kind of talked about those a little bit. But one thing you can see is because of the Armed Forces Medical Examiner support to our federal brethren, AFDL supports those individuals as well, those different agencies. So the Department of State, the Peace Corps, FBI, NASA. We assist in identification efforts whenever there are decedents associated with those events. So you can see on the left, there's actually, that is a picture of us doing a recovery mission in Haiti following the earthquake there. This instruction is what gives us our authority to perform identifications for both the past and the present accounting missions. I'm not going to go into detail, you can read it yourself, but do know there is statutory guidance out there that speaks to that. Regarding our personnel, if you look, this is an older photo, but you look at them, you'll notice something very unique. Do you see anybody in uniform in this photograph? There's one person, it's Colonel Finelli, our former director, he's hiding in the back. But we have a staff that's entirely contractor-based, and they are highly dedicated to this mission. And they're extremely educated. They're very, very smart folks. They have a lot of schooling. If you look, we have almost 80 master's degrees in our division of about 150 individuals. We've got 3 PhDs. You almost have to have an advanced bachelor's degree or master's degree to do DNA work. It's extremely difficult. I mean, honestly, it's kind of amazing I'm even in charge of it. I'm a forensic anthropologist by trade. I got a C in genetics, and my folks are over there. The first time I took the class, so you know, it shows how difficult it is, right? But we are also an accredited laboratory. We are accredited through the American Society of Crime Laboratory Directors, through the FBI, Quality Assurance Standards, and we have a DoD oversight committee that ensures that the DNA work we are doing is accurate and that we are reporting out the correct information. Now I want to go into just a little bit of the other missions before I focus in on the past accounting mission. Our nuclear DNA section supports the medical examiner and our current conflicts. They also do this criminal law enforcement work as well. So you can see we were most busy in that section in 06-07. That was the height of the conflicts in both Iraq and Afghanistan. And a lot of what we were seeing was blast injuries, improvised explosive devices that led to a lot of body fragmentation. And so we were trying to identify as much of our service members as possible, and that requires a lot of DNA testing because you get severed limbs, unfortunately, and other parts that you can't identify by other conventional means like dental identifications and fingerprints. And so a lot of that responsibility falls on DNA. So you can see our numbers, and we're very happy to have a decrease in our workload in our nuclear section, have actually decreased significantly since the Fallujah time period. Some of the notable missions that our folks have been involved with, you can see here the USS Cole, both the Columbia and the Challenger space shuttle tragedies. We assisted JPAC with the Hunley and the Monitor identifications, or at least attempts to identify those individuals there. And you can see these other ones. One fact that a lot of people don't know, yes, we assisted in identifications for individuals in the Pentagon, but we also, during 9/11, assisted in the DNA identifications for those victims of the Shanksville crash as well. And we assist the National Transportation Safety Board with a lot of commercial aircraft crashes. So there's a lot of different missions that we get involved with beyond just the Department of Defense. We have an emerging technologies group. We're not allowed to call them research solely because of the type of money that we utilize, but they're doing research. And that's my opinion, okay? And what they're doing is these are the folks who are constantly pushing the envelope, trying to assist in identifying your loved ones, especially like cracking the punch bowl and whatnot. We have a very synergistic relationship between the different sections within our group, within our division, and what one group does is going to really affect the organization as a whole. So it's very critical that we do these. Some of the more notable cases, those of you might notice the picture on the bottom right, that's the last Russian Tsar's family, the Romanovs. We assisted in the identification of all of them, including Alexei and Anastasia. Yes, we identified Anastasia. There is no Anna Anderson. At least there is an Anna Anderson, but she's not Anastasia. We also assisted in the identification of the Tomb of the Unknown, Lieutenant Michael Blasey, from the Vietnam era. So we've been involved in a lot of breakthrough technologies to assist in the identification effort. What they've also done is they have also created a mitochondrial DNA database. And what helps us do, what makes this so beneficial, is that if we can't identify a set of remains, they can look at the information and usually tell us approximately where the ancestors of that individual arose from, what geographic location they are from. So if we can't tell Dr. Holland that we have a U.S. service member, we can say that person was likely of Asian origin. And if they're coming from some of these older conflicts, there were very few Asians who actually served in the U.S. military during Korea or World War II, for instance, a better example. And so sometimes we can use that to exclude somebody from that identification process. I'm going to talk a little bit about DNA technology because I know we have some new folks here. We utilize primarily 3 different modalities, 3 different types of DNA. You've got nuclear DNA, your autosomal DNA, you've got your mitochondrial DNA, which I think most people are familiar with, and you've got what are called Y, short tandem repeats, Ys being the male chromosome, the male sex chromosome. Your nuclear DNA is what's at the center of this cell. It's essentially the brain of your cell. You get half of your nuclear DNA from your mom, and you get half of it from your father. And this is what makes an individual you. This is what we use for current conflicts to identify individuals, because no 2 people are going to be the same as far as nuclear DNA goes unless you have an identical twin. One of the problems with this is you only have 1 nucleus per cell. So there's not a lot of repetition as far as that goes. Your mitochondrial DNA, on the other hand, is passed from mother to child. And what makes this really useful is that there's very little mutation. It's going to be the same from mom to child. And if you look at all of the little mitochondria, you've got a lot of them in your cell. The purpose of a mitochondria, it produces energy. It's the power plant of the cell. And so you've got a lot of that to produce that energy. There's a lot of redundancy, and it tends to survive a little bit better. The way it's passed from mom to child is that there's mitochondria in the egg, and yes, there's mitochondria in the sperm, and we're working basic biology here, but all the mitochondria in the sperm are located in the tail. That's what's used to power the tail to push the mitochondria as it's swimming through to fertilize the egg. So once the sperm penetrates the egg, that tail falls off, and all the father's mitochondria stays with the tail. It doesn't penetrate the egg, so that's why you just have mom. And then you've got your Y STRs, and that's passed from father to son. So this shows you the chromosomes. You've got 23 pairs of chromosomes. Again, half from your mom, half from your dad. You can see there's a little Y one. It's a little short, stubby one compared to the X. That's what gives guys, your ability to find everything and not have to ask directions when you're driving. That's good for your wives. Yes. Again, my opinion. But we're also starting to look at X chromosomes and what you pass from both parents to your daughters, and we're starting to look at that as well to assist in identifying individuals. So when we're looking at that nuclear DNA, this is when we're going after your siblings and your children and a spouse. This is what's going to give us this autosomal DNA. And I talked about some of the advantages of using these and some of the disadvantages. This is highly discriminatory. We can usually individualize someone if we can get a full profile from them. And the problem, though, that we see is that it becomes diluted as you move out generations, because you're getting half from mom and half from dad. So every time you go further away from that individual on the family tree, they're going to be less and less related to that individual. So it's more and more difficult to use nuclear DNA, and the statistics become less and less. We talked about the mitochondrial DNA going from mother to child, so it doesn't matter if you're a male or a female, you will have the mitochondrial DNA of your maternal line. And again, lots of copies, very few mutations, and it survives pretty well. That's why we go to this primarily when we're looking at these older remains. And your Ys, again, from dad to son only. And I know this is an eye chart. A lot of you have seen this. I see people in the audience going, oh my goodness. All we're trying to get at is when you look at a family tree, your YSTRs are what are in green. Your maternal references are all those ones that are in red. So a lot of them are going to be on the left-hand side of the chart. You can see we have a lot more options with our mitochondrial DNA. We've got fewer with our Y. And then when you look at the nuclear, they're on the bottom for the most part, and a little bit the SIBs that are up there by that missing individual. So fewer for that. But they're all very important because mitochondrial DNA and YSTRs, they're not individualizing. So you can have multiple individuals in this room who could have the same mitochondria and mitochondrial DNA, but they might not necessarily be related as far as you know. In fact, in Caucasian males, or just Caucasians in general, there's about 7 to 10% of us who all share the same mitochondrial sequence. So then we have to go and look at other avenues of identification. And if we have our Ys and if we have our autosomals, then we can use those to start excluding people or potentially match them. So I want to talk about our past conflict accounting. I'm going to embarrass somebody. Susie, please stand up. Miss Susie Barrett Ross, who's over here on my right-hand side, she is the woman in charge of this entire operation for our office. And her staff is sitting at the table with her. And I would just like everybody, if you would, please give them a little bit of applause. This mission for almost 20 years. So while Mr. Panic, he's kind of the heart and soul, I think, of AFDL, I think Susie, she's pretty much the brains and the brawn behind our operation. You know, and they're an extremely dedicated force. I told you that they're all civilians, they're all contractors actually, but they're just as patriotic as somebody who wears our uniform. They're just as dedicated to the families and our service members as anybody who's ever served in the military, and many of them have served in the military and are veterans themselves. So this is the type of condition of remains that we would get on a good day from Dr. Holland and JPAC. And these are some of the statistics I'd like to show you. This is what we're actually currently doing. This year we set a goal for ourselves to process 1,300 samples, so 1,300 different pieces of bone or teeth or other samples that Dr. Holland could provide us. And we've already surpassed that for this fiscal year, and that's all because of Ms. Barrett Ross and her folks. So we have increased our efficiencies, and we are totally kicking some booty as far as producing some output here. You'll see a slight dip last year. That's because our entire office moved from Rockville, Maryland, up to Dover Air Force Base, Delaware. So while we were still accomplishing the mission, we had to ramp things down a little bit while we moved all of the equipment, we moved all of our references and our databasing up there. So we're doing a phenomenal job. And we're going to probably hit around 1,500 at least, I think, by the end of the fiscal year. So you can see here, these are the specimens that were submitted by JPAC by conflict. For this fiscal year already, it's about 32% of the cases that we processed are from Korea and Cold War. So it's a fair amount of the work that we're doing. As far as family references go, I cannot stress enough how important you all are to the identification process, because without your family references, we would never be able to identify your loved ones. So I highly encourage you, if you don't know if you've provided a sample or if you're a good candidate, please stop by our table. We're We're right out by the elevators and check. We've got a master list and we can tell you if we've got your samples on file. But you can see right now, we've got about 22,000, a little bit over that, family references on file with our office. And we've received about 1,300 this year thus far. If you look at these samples by conflict, you can see the Korean War. We've got about 88% of you represented, or your loved ones represented, which is really incredible. You by far have contributed the most DNA to our effort. So I applaud you. Oh, and by the way, we do not take blood samples. It's all just little cheap rubbings, so if anybody doesn't like needles, you don't need to worry. It's very, very painless. For Cold War, you can see we're at 81%, so it's still absolutely incredible. So we're doing really, really well in that respect, but we could do a little bit better. And I know we've got some folks that we've been looking to target, if you will. It's not in a bad light, don't take that wrong, but we would love to get your DNA, if you don't mind. We won't use it for nefarious purposes. We do not clone people or anything like that. Right, if you look at the named comparison reports, these are the reports that we send out to JPAC. Please keep in mind, we're not doing the identifications. We are a service laboratory for the SIL, for Dr. Holland. We provide him all of our DNA results, and he is going to analyze that and his folks along with all of the other contextual information, and the identifications will come out of his office. So we have sent out about 136 named reports, and you can see quite a few are from the Korean War. But I do need to preface this that some of these are subsequent identifications. They're not necessarily all new identifications. So if we get remains in, and for instance, a lot of you know about the K208, you've got a lot of commingled remains. When we get subsequent identifications, we're still reporting those out, but they don't count towards our new identification numbers. So this really ties into these different points to consider. Just because we get a sample in doesn't mean we're necessarily going to get an ID if we are able to get DNA from that. And that is because of a variety of factors. Well, one, if we don't get DNA, it's because it's too degraded, but it doesn't mean we're not going to keep trying. We keep pushing the science, pushing that technology, looking for how can we get DNA out of these remains. You have commingling issues, you also have these subsequent recoveries, and sometimes these individuals may not be U.S. service members. For instance, in the Korean War, this was a United Nations effort. You have local nationals, you have third country nationals, you have people from Turkey, from South Africa that could potentially be commingled with our U.S. service members that we don't necessarily have references for. We do have some references for some Australians, but we don't have it for everybody involved in this conflict, obviously. Some cases we don't necessarily have family references for, and in some circumstances we get tiny, tiny bone fragments, and that's all we may have. And no matter how good of an anthropologist you are, if you only have a fleck that's that big, sometimes it's really difficult to tell is it human or it's not. But we have DNA tests that will actually speciate that. So we can tell you it's a cow, it's a pig, it's a goat, or it's a human. And so that is one of the tools that we have in our toolbox. And then we come here, and I talked about the family references. Never, it's not a good photo op when you're taking a cheek sample from somebody. It's almost like when you get somebody who's eating. They just, well, you all are very attractive. This is just not probably the best light to capture you in. So we'll keep going. All right, what can we get DNA out of? We can get it pretty much from any biological samples. A lot of people just think about blood and saliva, cheeks, maybe some fingernails, although fingernails are pretty tough. We can get it out of hair. We've gotten references, alternate references in from families such as hearing aids, and we've been able to get it off of the hearing aids and the wax. We have found hairs embedded in eyeglasses, for instance. We have found them in biopsy specimens. So somebody goes in and they have a test done at the hospital and they're holding a piece of tissue. We have actually been able to take, that's a paraffin block that you see on the left-hand side. We've been able to take a little piece of that specimen that the hospital retains, and we've been able to get DNA out of it. And then as far as has some of these other type of references. You've got razors, toothbrushes. You always have to be careful because other people can use your razor, hopefully not as much your toothbrush, but there have been cases where we've had multiple users of the toothbrush. Different types of clothing items, things like baseball caps, we've been able to get DNA off of. So if you've got we've had people submit baseball caps from family members that they had. Although sometimes it's difficult to confirm that only that one person ever wore that piece of clothing. We've been successful in getting DNA out of watches. Sometimes if you have like a metal watch band, and you know you'll get the little scuzzy gunk from your skin cells in between it, it's really hard to clean out. We've gotten DNA from that as well. So these are all things that we could potentially use. Now, originally, I was going to kind of go into some of the cases that Dr. Holland was going to speak to and talk about the DNA associated with that, but I'll talk about those case samples and you'll get more of the contextual information in your next briefing. I think some of you are very well aware of the problems that we've encountered with the punch bowl samples from the Korean War. So the National Memorial Cemetery of the Pacific, we've got several folks, we've got over 400 who are in there from the Korean conflict. And the problems that we've seen are we just can't get DNA out of these remains. They look good morphologically to the eye, they look like they're really solid, but you can see they're kind of covered in a white powder. And this has been problematic for us. And the reason why is because these remains were sent to Kokura, Japan, and they were immersed in vats of formaldehyde. You're talking 48 to 50% formaldehyde. Now, my background is current conflicts and working with the medical examiner. We use 10% formaldehyde to preserve specimens. And we can't get DNA out of that. You're talking almost 50% formaldehyde, and then you add a hardening compound on top of that has additional chemicals that will inhibit your DNA, and you're talking about a real head scratcher. So these were immersed for an extended period of time. They were allowed to dry, and we have just not been able to get good results out of these. We did recently, though, get DNA out of a tooth from a punch bowl Korean veteran who was unknown, and we sent the information to Dr. Holland. But you can see we tried first with a piece of bone, piece of the right femur. We were unable to get good DNA out of that, so you can see a little piece of the bone, but we were able to get DNA out of the tooth. Now, some of you might have a hard copy handout of this presentation. There's actually another bullet in the hard copy, and it's not true. We haven't been able to test it with the next generation sequencing yet. It's in the queue, but we haven't gotten anything out of that. So now that individual we've been able to identify. And this is one of the first solid identifications we've been able to get with DNA out of the punch bowl. So this was a disinterment. Another was from a unilateral turnover, and I think a lot of you are aware of K208. So these were remains. We received 208 boxes. Supposedly there was one individual in each box, but as the SIL quickly found out, that wasn't necessarily the case. So in this instance, we received 121 samples from the SIL between 01 and 12, so for over a span of 11 years. So this is how long we will pursue this advanced technology. I told you technology is changing all the time. We went through different protocols during this time period to try to crack this nut. And what we found is that we had 11 bones that shared the same mitochondrial DNA profile. And that was found in 7 different boxes. And you can see the little bones that are colored in there. So little pieces of the same individual were found in a variety of different boxes. This is a case of severe commingling. But this is going to show why those alternate or those additional family references are really important, because of those 11 samples that had the same mitochondrial profile, when we went back in and compared against the family references, we had 3 different service members who had that same mitochondrial DNA profile. So we couldn't identify this person yet. So what we did is we ran the YSTRs. We looked at the paternal references and we compared it against them. Now, while we didn't have paternal references for all of those individuals or got results from all of those samples, we were able to exclude one of those three. So now we're down to 2. So what our folks did is they then went and ran those autosomal, those nuclear tests on those. We were able to get results from 2 of those specimens, and one of those, even though it was a sibling, and so we don't have a 100% match, but we had enough of a match that we were able to say that this person statistically appears to be this individual, one individual, and send those results on to Dr. Holland. And then our third case is from the joint recovery operation that occurred in the Chosen in 2004. Again, this was another long-term project for us. We received 103 samples over 7 years. We tested all of those for mitochondrial, and we tested some for our Ys. You have to keep in mind the YSTRs and the autosomal don't work as well for these degraded remains, and we're still working on those technologies. But what we got is 30 individual mitochondrial DNA profiles from 103 samples that were sent in. So it's a lot of different people that we have to weigh through. But fortunately, we had a brother who submitted a reference. And what you can see here, these are all the samples that we received in, all the different bones from all the different time periods. And the ones in red were the ones that were identified to our single service member. So again, they're not all in the same batch, and they're spanning a long time period. So you can see what we're talking about with these subsequent identifications. But we had a brother for this individual who provided us a reference, and we were able to get a match for him. So we were able to send this individual home, and he was buried. It was identified in October of last year. So what this shows is this is showing our sample success rate. Right now, a lot of people think, oh, we're having a lot of problems with getting DNA out of remains in general. Let me tell you, we have over a 90% success rate with mitochondrial DNA. Over 90%. Not including the punch bowl. Well, that does include the punch bowl, but the punch bowl is our biggest problem. But you can see these are different modifications to protocols, different things that we brought online. And the 2 at the bottom are our YSTRs and our nuclear. You're going to say, well, your success rates are going down. Well, just like with the SIL, and when you go and do the recoveries, you pick the samples you think are going to work the best first, and you do those, and then you start working on the more difficult ones. And that's why our success rate has gone down, because now we're back to our more degraded samples. But we brought some new protocols online that we think are really going to start boosting these numbers up, and we're expecting our success rates, specifically with our Ys, with our paternal references, to be about 40%. So one of those protocols that we brought on, we call DMIN, Demineralization Protocol. And this is what you see here in 2007 that is going to bring up that success rate. And this is something you're probably going, well, what the heck is demineralization? What does that do? Essentially, these are the chemicals that we are going to put into the bone powder after we crush that bone up or that tooth, and it helps to break apart that bony matrix so we can get at the DNA inside. And that has revolutionized this science in general. This is now a technique that is implemented worldwide in the civilian and in the federal sectors. And it has reduced the amount of bone that we have to have, that SIL has to provide us, and it's increased our overall success rates. And we just brought our second iteration of this online in July. And so this should further increase our ability to get DNA out, and we've been able to combat some of those inhibitors that are keeping us from getting the DNA. So this shows you kind of what our success rates were with different types of bone. The bones in the legs, the long bones, always have been the most successful because that has the thickest cortical bone, or the outer layer of bone. So this is before that first demineralization protocol. once we started using the DMIN protocol, you can see our success rates for all those different types of bones are now roughly equal. So it doesn't matter what type of bone Dr. Holland presents to us, we have a pretty good chance of getting DNA out of it. And some other recent advances, we have just brought online a low copy number. So little bits of DNA for those Y shorthand of repeats. We brought this online. We are expecting this to increase the success rate. I've talked to you about that second demineralization protocol. We're also looking at new areas, new kits to bring online.